now let's talk about the experiment. This is the process that we use to test the hypothesis. In order to do the experiment, you need to make predictions of what you think is going to happen. So you need to write your hypothesis. You need to determine your variables, so everything that you can use or will be affected by this experiment. You need to create an experimental procedure, and that is so that you can do the experiment, and then you need to carry out this procedure. So, making predictions. These predictions are going to be based on your experiment that was created to test your hypothesis. So, when you're making predictions, you should use if and then statements. So, like, if the hypothesis is true, then the results of the experiment will be. An example is if exposure to fertilizer increases the reproduction of bean plants, then the number of pods per plant should increase in plants that are exposed to fertilizer compared with the control plants. Determining your variables. A variable is the factor in the experiment that is being tested. So there's 3 different types of variables. We've got the independent, the dependent, and the control. A valid experiment only has one variable, so one thing that is being tested. The control variable, this is what is kept constant, so it doesn't affect the outcome of the experiment. The control is what you're going to use as comparison to what you want to test. The independent variable, this is the variable of interest. This is what you're going to change on purpose in the experiment, and you're only going to change one variable at a time. So you're not going to try 2 or 3 different things first. If you're starting off an experiment and you want to see, let's say, how light affects the growth of plants, or what different factors can affect the growth of plants, you're going to start off with, let's say, light first, instead of trying to see if light and water are going to affect how the plant grows. And so only changing one variable at a time allows you to know which factor will affect the dependent variable. And the dependent variable is the variable that you're measuring. It's the one that you're observing during the experiment. So this is what is being changed involuntarily and is being affected by what you're doing with the independent variable. So let's do some examples. So an example would be exposure to fertilizer increases the reproduction of bean plants. Well, the independent variable, what we are changing, is the fertilizer. The dependent variable, so what is going to be affected by us changing the fertilizer, is going to be the number of bean pods that are growing on the plants. And the control variable, the thing that doesn't change no matter what, are other things like water, temperature, and light. So now let's try another. If you want to go ahead and try this on your own, now would be the time to pause the video and see if you can figure this out, then play it whenever you're ready. Alright, so the relationship between plant growth rate and duration of light exposure. So which is the independent, dependent, and control? Well, the independent is, remember, what we're changing, so that should be the amount of time of exposure to light. So that you are changing how much time you're letting each plant have access to light. The dependent, the thing that's being changed because you're changing the light, is the plant growth rate. You're affecting how the plant is growing just by changing the light. The control is going to be all other aspects of growth. Again, this will be water, soil, food, anything else that you need to help the plant grow. Now let's talk about designing a procedure. Procedures need to be step by step on how the experiment is going to be completed. A good procedure is going to be one that someone on the other side of the world can complete just by reading your procedure. So everything you do, you should be writing down and in some way, shape, or form turning into a procedure. And generally with procedures, you're going to want to use quantitative methods because they're more precise and not as subjective. Quantitative methods are numerical methods. They always use numbers. Whereas qualitative, these are descriptive. So you could, if you're doing an experiment, let's say you're using hydrogen peroxide to water some plants and not others. So a quantitative method, a numerical method, would be measuring the growth rate or measuring how tall these plants get after a certain period of time. A qualitative method, the descriptive method, is you could talk about whether the hydrogen peroxide is affecting the coloration of the plant. So you could say, well, the one that's being watered with hydrogen peroxide all the time is a light green color, whereas the control is a deep green. Well, the problem with qualitative is you may think something is a light green color, but I could think it's a medium green color. So there is some subjectiveness into using qualitative descriptions, whereas numbers are numbers. Nobody can dispute them. And replication. So you're going to always want to do more than one trial. The more trials you do allows you to collect more data, which gives you better experimental results. So if you did 2 trials of something and they didn't match up at all, then you don't really have a good representation as to whether your experiment worked or not. But let's say you did 5 trials of this experiment and 4 of your trials matched up pretty similarly, but 1 of your trials was kind of way out in the ballpark. Well, you call that the outlier. You throw that one out because the majority of the trials matched each other. And so also doing more trials allows you to see that your experiment can be replicated and you can get the same results each time. Now let's talk about data. These are your results from your experiment. Your data should be organized into some kind of chart, table, or graph so that you can see your data better and it helps you analyze your data. So if your results from your data match the prediction, that means your hypothesis is supported. If they don't, then that means it's falsified, which means you're proven wrong, which means your hypothesis was not correct. And the conclusion. The conclusion is where you're going to put your answers to your hypothesis based on the data that you've collected from your experiment. So this is where you're going to write down whether your hypothesis was right or whether it was wrong. So if your hypothesis was right, then what you should also write in your conclusion is additional experiments that you can do to further the support of your hypothesis. So let's say fertilizer does affect the bean pod production in a plant. Well, the next thing you could do is increase the fertilizer because you know that works, or leave the fertilizer the same and see if maybe light affects it. But if your hypothesis wasn't supported, then the other thing you should be writing in your conclusion is alternative hypotheses for testing. So let's say fertilizer didn't affect the bean pod production. Well, then maybe you can say, let's test temperature next time. So maybe a higher temperature is going to increase bean pod production. That would be something that you would write in your conclusion. So some examples for supported hypotheses. Exposures to fertilizers increased the reproduction of bean pods. The more bean pods were found on plants when there was more fertilizer added. So a conclusion would be we could try different amounts, concentrations of fertilizer, or you could try different brands or types of fertilizer, or you could also see if a combination of variables affect the replication. Like you could try different combinations of fertilizer and water or fertilizer and light. And here's some examples if it's not supported. So the exposure to the fertilizer didn't increase the bean pod production. The bean pods, there were the same number or less bean pods than as the control. Your conclusion would say, well, we could try testing other factors like temperature of the air or soil, the amount of water the plant receives, or the amount of light that the plant is being exposed to. So, what you write in your conclusion is dependent upon whether your hypothesis is correct or incorrect. And then the final part of our scientific method is the retest. So in order to verify your results in your experiment, you'll need to be able to retest your experiment. So if your experiment gets retested and the same data is found by another person, then your experiment could eventually become a scientific theory. That takes a while. It takes a lot of people to do the experiment and figure it out, but it could happen. So you need to make sure that your experiment is retestable. It is able to be done by somebody else. And if you do it on 2 different times, let's say you did it one day and then like 3 weeks later you did it again, you need to make sure that you can get the same results. Alright, so let's just do a little bit of a recap. So the importance of the scientific method. While observing something that is what's going to pique your interest in the scientific method, then you've got to make the hypothesis that you can test, and your hypothesis is going to be based on whatever you observed. And then you're going to create the experiment that is based on your hypothesis. Then you'll make a step-by-step procedure so that people can replicate your procedure so that people can replicate your experiment someday. And then you're going to record your data in some kind of table, chart, or graph. And finally, you're going to write a conclusion that explains what happened in the experiment and where you can go from there. So this is the end of the lesson. Write down any questions that you have in your notes so that you can submit them for your journal, and I will get back to you after I read your journals. If it's something that you need clarification on before you go ahead and do the worksheet, please send me an email or text me. That's fine. And have a great day.